Okay, well, thanks very much. It's a real pleasure to be here today at this research symposium, and also just to be part of the school and to be conducting research here. As you can see from all of the presentations, there's just a huge array of research being done and very inspirational faculty, staff, and students. So I'm John Marshall. I'm an assistant professor in residence in the divisions of Biostatistics and Epidemiology in the school. And I'm going to be talking today about designing safe strategies to eliminate mosquito-borne diseases using gene editing. And I lead a research group which specializes in the mathematical modeling of infectious diseases, in particular mathematical modeling of mosquito-borne diseases, including focusing particularly on malaria. We have a group of computer scientists, genomicists, applied mathematicians, epidemiologists, computational biologists, and biostatisticians, postdocs, graduate students, and undergraduates. Together we develop mathematical models of the transmission of these mosquito-borne diseases. And the focus of our work is currently on the use of gene editing techniques to control those diseases. Sean Wu, who's a PhD student in my group, is going to be talking about malaria models after me. And I want to start with this slide. For me, it's a big inspiration for why we are working on gene editing to control these diseases, in particular malaria. It's really difficult to eliminate malaria from the African continent. When I was doing my PhD a little under 10 years ago, there was a death from malaria about once every 30 seconds. And today, with the widespread use of insecticide-treated bed nets and insecticide treatment of walls and houses, that number is about 1 death per minute. But that's still a huge amount of deaths across the continent, and there's been a huge distribution of bed nets and also anti-malarial drugs across the continent. What's shown in this figure in yellow and orange is the regions where mathematical models predict that we could eliminate malaria from these areas using insecticide treated methods, so the bed nets and the spraying of walls. And then in the green and red, these areas here, it's predicted using mathematical models that we could eliminate malaria using some combination of insecticide treated nets and walls and anti-malarial drugs, which would be distributed throughout the entire population. And then this remaining portion in gray is the majority of the area that's affected by the disease, and we do not predict using mathematical models that we can actually eliminate malaria from these regions using the currently used and available tools. And this is actually, the significance of this is huge, because in these models we're actually assuming that 90% of people are using both forms of interventions in these areas. And we're also assuming that they work as perfectly as they did in the experimental trials. We're assuming that there's no insecticide resistance. We're assuming no drug resistance. And even with those highly optimistic models, which are actually unrealistic, I mean, it's very difficult to reach these areas due to social unrest, then we still predict that we couldn't eliminate with these tools. So that's why we have an interest in gene editing and other technologies. It's not something that we would start off with just off the bat. It's something that is motivated by the inadequacy of the current tools. So the goal is of this technique is to either have mosquitoes that can't transmit malaria, or to just get rid of all of the mosquitoes that would transmit the disease. And there has actually been success made in engineering mosquitoes incapable of carrying malaria and transmitting it to people. There's still a bit of work to be done in that field. And there's also been some work in engineering mosquitoes that are infertile and therefore unable to have progeny. What is needed, so engineering one mosquito that can't transmit malaria might be a Nobel Prize, but it's not much use in malaria control because you need to make all of the mosquitoes across the continent unable to carry those diseases. So actually a development at UC Berkeley, so Jennifer Dowder and colleagues have engineered a way that you can precisely cut a site in a genome and choose which site that's going to be, and we can use that technology actually to drive a gene into a mosquito population. It's very interesting science. Actually, the bacteria, it's their defense system against viruses, and because bacteria are single cells, they need to fight. They don't have immune cells specifically, so they actually have these genomic components that can cut the DNA where the virus is and then remove it from the genome. And in this case, what we're doing is we have a gene that might confer refractoriness to malaria, and we cut the homologous, or the opposite chromosome, so you'd get one from your mother and one from your father. If you have this gene from one of your parents but not the other, it'll cut the opposite chromosome, and then it serves as a template for DNA repair, leading to, if you had one copy, now you have two copies. That biases inheritance in its favor, and it spreads into a population. You can either spread a gene conferring refractoriness to malaria, or you can spread a gene that would disrupt female fertility. This is due to the incredibly impactful discovery of Jennifer Doudna and colleagues. So this technology, genetic engineering, is highly controversial, and so we've done work where we've spoken to people in communities and asked them, what they think about this technology. And they've said, as an example, I would have to see an example of modified mosquitoes reducing malaria in another village before I believe this claim, or you have to start somewhere. From this, people will know whether it's a good idea or a bad idea. I would like you to conduct a trial in my village, because I would like to be an example for another community. So this has inspired our interest in whether we can conduct a confined field trial of this technology, so whether we can release in one village, have modified mosquitoes there, and without it spreading to the neighboring village prior to the wide-scale release. And we use mathematical models to address these questions. And mathematical modeling, which is the work in our group, is really how we integrate data from multiple sources. So we have data on the inheritance pattern of these systems, the life cycle of the mosquito, the landscape. So this would be a number of households and a number of households in another community connected by movement of mosquitoes, and then the epidemiology of the disease that we're interested in. And I'll just finish with a model implementation. So here we're going to see a release of mosquitoes carrying a gene drive system in this community. And this community, there's no release. And so here we have a release. In the yellow, you can see the released transgene, and it's taking over this population. And then there's movement of mosquitoes into this community at low level, but because it's at a low level, it doesn't take off. And then we have a corresponding release of wild mosquitoes, which dilute the system out of this population and restore the wild type. And we hope that through mathematical modeling, we can inform the field trial design of this technology, which ultimately will allow its use on a wide scale to control these diseases and to reduce the impact of these disease burdens across the African continent. Thank you.